KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, good morning to all. So in this video we are discussing an overview of cell. In this video we are discussing overview of a cell. So overview means entire information of the cell. Means what are the outermost protecting layers, and so what are the matrix, or what is the cell constraints are present, so what is the importance of cell. Likewise, so each and every minute point of a cell also we will discuss. Now listen carefully. Cells differ greatly. Continuation. So in a previous video we have learned, suppose if you take a plant cell, outermost layer is a cell wall, and if you take animal cell, outermost layer is a cell membrane. So likewise we have seen some more information, isn't it? Next one, there in eukaryotes, if you see there developed nucleus is present. Developed nucleus is nothing in the sense of what is the meaning. It means the presence of nuclear membrane. Presence of nuclear membrane around the nucleus. Later all kind of cell organelles are present. So in case of eukaryotes, likewise next one in case of prokaryotes, all kind of cell organelle, all kinds of cell organelles are absent except ribosomes. So likewise we say. So along with those important points, so in previous video one more point also. So ribosomes are amembranous cell organelles. Amembranous means non-membrane. Non-membrane cell organelles, and definitely these are all present in both prokaryotes as well as eukaryotes we see. Later in animal cells, one more amembranous means non-membranous cell organelle is there. That one we will be taking as a centrosome. Likewise some other information we see. In this video, so what is the information we are learning now? That is cells differ greatly in size. One more time, cells differ greatly in size, shape and activities. Okay, so when the particular cells differ in greatly in size point of view, shape point of view and activities point of view, when a difference is there, so definitely there we will be differentiating. So for differentiating purposes point of view, definitely use some. For example, if you take there mycoplasma. The mycoplasma is smallest cells. Mycoplasma is the smallest cell. This information in previous video also I have given. And only 0.3 micrometer in length. Mycoplasma, if you see there, it is the smallest cells, and only the length of mycoplasma is just 0.3 micrometer. 0.3 micrometer. Next one, bacteria point of view. Bacteria could be 3 to 5 micrometer. Okay, means suppose if you take a mycoplasma is a smallest cell, and that one is the length is 0.3 micrometer in length, whereas bacteria cells, if you take there, the length or the diameter is 3 to 5 micrometer in diameter. Next one, largest isolated single cell. One more time, largest isolated single cell is egg of an ostrich. This is important. Okay, so largest isolated single cell. One more time, largest isolated single cell is the egg of an ostrich. Egg of an ostrich. among multicellular organisms, human red blood cells about or the size of the human red blood cells is about what it is 7.0 micrometer in diameter. One more time, once let us move back. Once you see mycoplasma 0.3 micrometer in length, bacteria 3 to 5 micrometer, ostrich the largest isolated single cell, and the multicellular organism point of view, in a multicellular or among the multicellular organisms, human RBC cells, human red blood cells are there, no? So what is the diameter of those cells is 7.0 micrometer in diameter. This is important, nalla save pannikonga. Next one, nerve cells. So nerve cells are some of the longest cells, nerve cells. So why? Because so definitely if you see in our body also nerve cells, so we will be considering as longest cells. So next one, the cells are cells also vary greatly in their shapes. So here we have seen sizes point of view, definitely differences we are finding. Likewise shape point of view also, cells also vary greatly. So vary greatly means definitely we can find the difference. Okay, so maybe disc like. So what are the shapes we will be finding here means maybe disc like, polygonal, columnar, cuboid, thread like, or even irregular. Once you see disc is there, polygonal is there, columnar is there, cuboid is there, thread like, or even irregular. Likewise there are different types of shapes of the cells we can find out. Okay, next one, the shape of the cell may vary with the function they perform. Means here one more time, once you see here, the shape of the cell may vary. Shape of the cell definitely chance is there to change with its function. So suppose depending on the function, so sometimes no chance is there to change its shape. Okay, point you understood, means depending on what, depending on the function, the shape of the cell chance is there to get change. I hope you understood this one. Now here somewhat information, a new information I brought for you, so listen carefully. That is this, RBC cells means red blood cells are there, no? RBC cells, what is the shape of the cells means, this 3 lines information is there, no? This koncham nalla understand pannikonga. So here what is information, no? RBC cells, red blood cells are there, no? Round and biconcave. So the cells, RBC cells shape is round and biconcave. Next one, white blood cells. White blood cells shape of the cells is amoeboid. Next one, columnar epithelial cells shape is long and narrow. Next one, nerve cell. Nerve cell is there, no? That one branched and long. Next one, tracheid. This is plant related, we have said, no? Tracheids, vessels, adida. Tracheids, elongated structures. Next one, mesophyll cell point of view, round and oval. One more time, once you see, RBC cells round and biconcave, white blood cells amoeboid, columnar epithelial cells long and narrow, nerve cell branched and long, and tracheids point of view elongated, mesophyll cells round and oval. So likewise here somewhat they have given different types of cells and its shape. This neat examination point of view important, but suppose if I give any extra points, no, you people are unable to take, that's why I am not giving extra points other than this, right? Next one, prokaryotic cells. Next information regarding prokaryotic cells. Now the prokaryotic cells are represented by bacteria, blue green algae, mycoplasma, and one more is PPLO. One more time. So under prokaryotic cells, what are the cells we will be taking? Bacterial cells are there, blue green algal cells. Blue green algae means cyanobacterium. Suppose any one of the names you can know, I think so you know very well. Okay, either blue green algae or cyanobacteria. Okay, next one, mycoplasma, and one more is PPLO. Here our textbook point of view, there are 4 different types of organisms they have been included under prokaryotic cells. இப்போ சரி இங்கே PPLO means what sir means once you see here PPLO is nothing but pleuropneumoniae like organisms. Pleuropneumoniae like organisms. Now they are generally smaller and multiply more rapidly than the eukaryotic cell. Means here what are the prokaryotic cells we will be taking as examples? No, under prokaryotic cell these are for examples at any cost you have to remember. Sariya. So first one say along with me, bacteria, blue green algae, mycoplasma and PPLO. Next one, they are generally smaller. These prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. Means here when compared to the eukaryotic cell, now prokaryotic cells are very small and multiply more rapidly. So for example, under bacteria, E. coli, if you take there, no, E. coli cell that divides within 20 minutes. Okay, so likewise within a fraction of means within a short period of time, so these prokaryotic cells multiply rapidly than the eukaryotic cells. Next one, they may vary greatly in shape and size. So already that point we have studied, but here why we are studying the shape and the sizes, definitely difference will be there greatly. Why we are studying? No, so author has given so 4 basic shapes of the bacteria. So here we are studying 4 basic shapes of the bacteria. So there bacilli is there, coccus is there, vibrio is there and spirillum is there. So this 1.5 line we know very well. Why? Because this information already we have learned in biological classification itself. So thereby but I thought to skip this one, but in a textbook also they have mentioned. Why should I skip? Okay, now the bacilli rod shape, coccus spherical shape, vibrio comma shape and spirillum spiral shape. Okay, now here cocci, bacilli, vibrio and spirillum. Likewise there are 4 basic steps. 4 basic shapes, not steps of shapes. Okay, so there 4 basic shapes of bacteria we will be observing. Ipo idi 2 lines koncham understand pannikongama, idi important. Now the organization of the prokaryotic cells is fundamentally similar. One more time you see there, organization of prokaryotic cells is fundamentally similar. Means here the organization means, suppose if you take one particular prokaryotic cells, no, these things must be there and these things condipa must be there and these things should not be there. Likewise and here arrangement must be like this. Likewise organization, everything in prokaryotic cells fundamentally similar. But even though prokaryotes exhibit a wide variety of shape and functions. So means fundamentally prokaryotic cells organization fundamentally similar. But here prokaryotic means even though fundamentally all prokaryotes or organization point of view fundamentally similar, but exhibit a wide variety of shape and functions. Wide variety of shapes and functions. Okay, so in this side, my dear students, what are the main important points we have discussed? Means now once you try to say smallest cell is mycoplasma, length is 0.3 micrometer. Next one, bacteria could be 3 to 5 micrometer. Next one, isolated largest egg cell is ostrich cell. Next one, in multicellular organisms, what is the diameter of the RBC cell? Means that is 7.0 micrometer. Next one, RBC cells shape is round and biconcave. White blood cells shape is amoeboid. Columnar epithelial cells shape is long and narrow. Nervous cells is branched and long. Tracheids elongated shape, elongated, and mesophyll cells round and oval. Next one, what are the examples of prokaryotic cells? Examples, what are the things? Bacteria, blue green algae, mycoplasma, PPLO. PPLO means pleuropneumoniae like organisms. So again what is the point? What are the prokaryotic organisms are there? No, all these are all very smaller and multiply very rapidly, more rapidly than the eukaryotic cell. Adi we know very well. Next one, so differences and condipa, definitely shapes and sizes, no, there are greatly vary. So there are different types of things are there. So there 4 basic shapes we will find. Okay, 4 basic shapes of the bacteria point of view. What are the things? No, bacilli, cocci, vibrio and spirillum we are observing. Now only we learn this one. So in prokaryotic organisms, that is all prokaryotic organisms organization is, even though fundamentally similar, prokaryotes exhibits a wide variety of shapes and functions. So that is the main point now we are learning in this particular class. Next one, all prokaryotes have cell wall. Once you listen carefully, all prokaryotes have a cell wall surrounding the cell membrane. So thereby here, first of all, what is a layer which is situated outside of the cell membrane? Cell wall. So once you see that all prokaryotes have a cell wall surrounding the cell membrane. Appadana cell membrane which is situated within the cell wall, or cell wall which is located outside of the cell membrane. Correct only now. But here once you see, all prokaryotes have a cell wall except in mycoplasma. So thereby now only we see no prokaryotic organisms, no there bacteria, blue green algae, mycoplasma, PPLO. Likewise we see no. Out of 4 types, no, one our knowledge point of view, one particular organism not possessing cell wall. Adi are mycoplasma. Mycoplasma nalli now you try to save this point in your mind. In a mycoplasma, which one is absent? Cell wall is absent. Cell wall is absent. Next one, the fluid matrix filling the cell is known as cytoplasm. Already this particular point in previous class also we learned. So means what is a fluid matrix, what is a liquid, or what is a matrix which is present in the cell, we will be considering as cytoplasm. So fluid matrix filling in the cell is known as cytoplasm. Next one, there is no well defined nucleus where in prokaryotes. So there is no well defined nucleus. Mean in previous class what is the point we learned? Well defined nucleus is absent, means nucleus is not surrounded by nuclear membrane. Point you understood. So thereby here, suppose a well defined nucleus means nucleus when it is surrounded by nuclear membrane, that is well defined nucleus. If no well defined nucleus means nucleus is not surrounded by nuclear membrane. Suppose nuclear membrane is there means well defined. Suppose if nuclear membrane is absent means no well defined. Simple only, no, nalla save pannikonga. Next one, once you see here, the genetic material is basically naked. Where in prokaryotic organisms, side-heading prokaryotic cell only, no. So that's why this entire information is prokaryotic cell information belongs to prokaryotic cell. The cell genetic material is basically naked. Which one is absent we are observing? So there is no well defined nucleus, means nuclear membrane is absent we are seeing now. If nuclear membrane is absent means definitely that particular genetic material we will be taking as naked. Naked. So next one, not enveloped by nuclear membrane. You see there? Now we will be getting off clear information. So there is no well defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. Next one, in addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. This is important. Listen carefully. In addition to the genomic DNA, now only we are seeing, no, suppose one particular organism is there, no, there definitely genetic material must be there. In addition to the genetic material or genomic DNA, many bacteria, so in so many members of a bacteria, no, so they have what? They have small circular DNA outside of the genomic DNA. Listen, did you listen carefully? Once you listen, so in many bacteria have circular DNA outside of the genomic DNA. Appadana here inge one. Totally how many types of DNAs are there? Rendu is there. One is genomic DNA and one more is small circular DNA. This particular small circular DNA we will be calling as plasmid. So means if plasmid now we will be considering as extra chromosomal. We will be considering as extra chromosomal genetic material. Appadana already one irukku. Next one extra irukku. So that's why we will be considering, we will be taking that one as how extra. Okay, now here one more important point. What is the point? So this particular extra chromosomal circular DNA only we will be taking as plasmid. Next one, plasmid contain or plasmid DNA confers certain unique phenotypic characters. Means if you see their particular, because of the presence of a plasmid DNA in a bacterium, so this particular DNA also it will do some kind of important activities in the cell, or important activities in the cell. So okay, so thereby now here what is the point we are going to take is so unique phenotypic characters. Next one to the bacteria, and one such character is resistance to antibiotics. Okay, resistance to antibiotics means here because of the presence of plasmid, so this bacteria showing which one? Bacteria showing resistance to antibiotics. Okay, so bacteria showing resistance to antibiotics. So here the bacteria showing resistance to the antibiotics because of the presence of plasmid. Okay, next one here, and plasmid DNA is used to monitor bacterial transformation with foreign DNA. So means here what is the point? One more point. So here there definitely this particular bacterial members DNA or bacterial members plasmid DNA we will use for the transformation of which one? Foreign DNA. Means here this particular bacterial cells plasmid we will be using. On which purpose we are using, sir? This one means definitely we will use this particular plasmid DNA for transfer of which one? Foreign DNA from one organism to another organism. Point you understood. So thereby here 2 points. One point, when this particular plasmid is present in the bacterial cell, that particular plasmid, because of the presence of a plasmid, so bacteria shows resistance to the antibiotics, and this what is a plasmid we are studying, that particular plasmid DNA we can use for the transfer of a foreign DNA from one organism to another organism. That is only we will be taking as, or we will be calling the process of transformation. So in a second year you will be knowing about this one very clearly. Okay, no worry, in a biotechnology chapter so very exclusively important points also you will learn there. No worry. Next one, nuclear membrane is found in eukaryotes. Nuclear membrane, that's we know very well. Why we are calling eukaryotes means next class EU means true. In eukaryotes, EU means true, karyo means nucleus, I said no. If true nucleus is present means what is the meaning? Definitely well-defined nucleus is present. Well-defined nucleus. Now here one more important point. What is the point? No cell organelles like ones in the eukaryotes. All are found in the cells except for ribosomes. So one more time you see that no cell organelles like in the eukaryotes are found in the prokaryotic cells. Means here except ribosomes. So thereby here what is the point meaning? Here all types of cell organelles are present in the eukaryotes, but all kind of cell organelles are absent. This no belongs to here prokaryotes. That's why either no organelles are present in the prokaryotes except ribosomes. So thereby in prokaryotes, what is the main important point? In prokaryotes, all kind of cell organelles are absent except ribosomes. So thereby in prokaryotes, how many number of cell organelles are present? Only one. That is ribosomes. Next information is prokaryotes. Prokaryotes have something unique in the form of inclusions. So here some kind of inclusion bodies or some kind of inclusions are present. So these inclusions are unique only in prokaryotes. Means these kind of unique inclusions we will observe only in prokaryotes and we will not observe in any other kind of organisms. Unique means that one special character. That's we know very well, unique meaning. Next one, a specialized differentiated form. A specialized differentiated form of a cell membrane is called mesosome. One more time. A specialized differentiated form. A specialized differentiated form of a cell membrane is called mesosome. So definitely mesosome. So this particular mesosome, from where it has been formed means it has been formed from the cell membrane. So cell membrane is differentiated and develops into mesosome. So the presence of mesosome, listen carefully, presence of mesosome is a special character or characteristic point of prokaryotic cells. Mesosome is there in any particular cell means that particular cell very blindly also we can take as a prokaryotic cell, and they are essentially infoldings of a cell membrane. Already we have learned this one. So these infoldings are definitely essentially what? Infoldings of membrane, cell membrane. Next one is cell envelope and its modifications. Cell envelope and its modifications. So there are also somewhat information to be discussed. Now most of the prokaryotic cells. Now one more time, most prokaryotic cells, particularly the bacterial cells have a chemically complex cell envelope. Chemically complex cell envelope. So suppose for example if you take there bacterial cell wall, if you take there their bacterial cell wall is made up of muramic acid or the peptidoglycans. Likewise there are different types of chemically complex cell envelope. Muramic acid is present. So mucopeptides are present. Likewise, so chemically compound or chemically complex envelope or chemically complex cell envelope we will be observing in bacterial cells. Okay, now here you see there cell envelope consists of tightly bound 3 layers structure. So once you see there, suppose if you take a cell envelope of a bacterial cells or the prokaryotic cells, if you take, what is the point here we can say? Definitely 3 layered or 3 layers structure we can see, and these 3 layers also tightly bound. நல்லா புரிஞ்சுக்கோ. இப்போ சார் இப்போ 3 layers are there no. First one யார் சார்? outermost one is பார்த்துக்கோங்க இங்கே outermost protecting layer is glycocalyx. And this particular glycocalyx are followed by the cell wall. Next one, this particular cell wall is followed by the plasma membrane. Now from outside to inside paathitingla, suppose outermost protecting layer is glycocalyx. We have written outermost protecting layer I didn't write, but through my voice you can get. So outermost layer or outermost protecting layer is glycocalyx, later followed by cell wall and then followed by plasma membrane. So this is a small information. Regarding cell envelopes and its modifications. My dear students, one important note, this is just starting of cell information, cell envelope and its modifications. In the next video, definitely I will continue with this. So now here in this video, what is information? Cell envelopes and its modification point of view we are learning. Suppose if you take like a cell wall or the cell envelope means 3 layered structures we are observing. Glycocalyx, cell wall and plasma membrane. In prokaryotic cells, mesosomes are present. These presence of mesosomes is a characteristic point of prokaryotic cell. How these mesosomes formation taking place? These are all formed from the infoldings of cell membrane. Next one, in prokaryotes, ribosome mattum the present, remaining all cell organelles are absent. Next one, here in prokaryotes, plasmid DNA, extra chromosomal, circular extra chromosomal DNA is present. So because of the presence of plasmid DNA in the bacterial cells, one particular point, so showing off, once you see there, confers a certain unique phenotypic characters. Phenotypic characters phenotypic characters. So what are the characters that we can see outside? Suppose one person is there, means we will calculate what's your height and what is your color, what is your hair color and what is your skin color. Likewise, what are the characters we will observe outside, means externally what are the characters we will observe, such kind of characters, our subject point of view, we are calling as a phenotypic. Now you learn this one. One more important point of our DNA, the plasmid DNA helps in transformation. Transformation means suppose if any kind of desirable genetic material, if you would like to transfer, if you would like to take from the one place to another place or from one organism to another organism, that particular process point of view in a second year, in a +2 definitely we will learn the help of plasmid. No worry. Next one, genetic material basically naked in a prokaryotes. Why genetic material basically naked means due to the absence of nuclear membrane. Next one here, in prokaryotic organisms, maximum members are having cell wall, but cell wall, once you see there, the cell wall is surrounding the cell membrane. That one definitely is present in most of the members, but in a mycoplasma, cell wall is absent. Here it is important point, and here 2 or 3 important points are there. And the entire information important only. So try to listen carefully. Suppose if you don't know anything, 2 times I have given explanation. So one more time try to watch. Easy only, you can understand clearly. Take care and be safe at home.